everyone, welcome back to Window Seat Studies. I hope you all are doing well. So, we were discussing about hallmarks of cancer, in which we have covered two important hallmarks, like how the tumor cells they are able to sustain a proliferative signal and evade the growth suppressor so that they can maintain their uncontrollable proliferation. Okay, and in today's lecture, we are going to discuss a very important hallmark other than those two, that is the property of cancer cell to be able to resist the cell death. So what happens is that when a normal cell it senses some DNA damage, or it senses that it is building up genomic instability inside a cell, so it has its own suicidal package which is triggered upon sensing those damages which are irreparable, so that this cell is better to be killed than to let it survive, because instead of accumulating all those genomic instability and affecting other cells, it is better to kill that cell. So in such mechanisms, the cells undergo they undergo the suicide. So that is very programmed, and that is why it is called the programmed cell death, because it is a very highly regulated pathway. And that pathway, we are going to cover that pathway in detail in today's lecture. So today we will be discussing apoptosis. So before discussing the mechanism of apoptosis, how it occurs, let's first discuss why do cell exhibit apoptosis and what it is exactly. So it is like a cell suicide. Okay, cell kills itself, but there should be a reason why a cell will kill itself. So the reason is that when a normal cell it is damaged beyond repair. So it happens when a normal cell is damaged beyond repair, or when it is mislocalized, or when it starts migrating abnormally, or when it starts proliferating abnormally. Upon all those circumstances, the cell decides that it should kill itself, because the DNA damage, okay, which can't be repaired, or the abnormal migration or abnormal proliferation, this can cause problems in the tissue. So for maintaining the cellular homeostasis, the cell takes the decision to kill itself, to suicide, and that is what we call the apoptosis. So it is a very regulated pathway, and that is why it is programmed, and that is why it is known as programmed cell death. So it's not like phagocytosis where other cell comes and kill that cell, engulfs that cell, but apoptosis is a mechanism which is initiated by the cell itself, and uh, uh, it undergoes the death. Okay, like the cell dies. So what happens is that first chromosome is degraded upon when cell senses one of those phenomena, like if if it has damaged DNAs, if it has double stranded DNA breaks in that, or it is migrating abnormally, or it is mislocalized, in any of those situations, it leads to the chromosome degradation. Okay, the chromosome degradation by caspases. The apoptosis is an enzymatic, like enzyme mediated pathway. So the enzymes that are involved are known as caspases. Oh, I'm sorry. It's called caspases. So those are proteases, okay? And due to the caspases, the chromosomes are degraded. How? Sorry. Well, they are proteases. So they are proteolytic enzymes, means protein degrading. So it degrade the proteins which protect the DNA, like the DNase inhibitors. Okay, we will discuss about those in details. So for now, just know that whenever cell senses any of those phenomena, it undergoes chromosome degradation. So first the chromosome is degraded, all of the cellular important cellular proteins, and they are degraded. Then cell undergoes cell blebbing, a phenomena known as blebbing, where where the like there will be some protrusions of the cytoplasms of the cell. Okay, like bulging of cell will occur. So this is a cell. So normal cell gets transformed into some structure like this. So those bulging structures, these are the protrusion. These are the bulging structures. Oh, oh, bulging structures. They are formed in the cell. So those protruding bodies are formed, and this is known as apoptotic bleeding. Okay, after cell bleeding, the cell undergoes fragmentation and it forms some bodies, the fragmented bodies, which are known as apoptotic bodies. So these apoptotic bodies, they are further engulfed by the phagocytes, so that this engulfment ensures that the subcellular components of the cell will not be released in the extracellular matrix. So the surrounding tissue will not have any subcellular components released in them. Like no heat shock proteins will be released, no DNA of that cell will be released in the tissue, surrounding tissue. So this will ensure that it won't provoke immune system, and as it doesn't provoke any immune responses, it will result into no inflammation. So apoptosis ensures that there will be no inflammation. So this is a very clear debris management, like whatever cell undergoes the degradation, but no subcellular component will be spilled in the surrounding tissue. So this ensures that the surrounding tissue won't undergo inflammation. So this will not elicit any kind of immune response. So that is why apoptosis is a very clean crime. Like the cell undergoes suicide in a very clean manner. Like it doesn't induce any kind of inflammation. So we have learned the basics of apoptosis. Like the Now let's learn about the mechanism. Okay, so just like apoptosis is a cell suicide, there is also one more thing when the cell undergoes death, which is known as necrosis. Okay, let's discuss about the differences between necrosis and apoptosis. Well, apoptosis is a very programmed, like it is a very programmed procedure. So this is a highly regulated pathway where cell undergoes a signaling, and then it undergoes the procedure to kill itself. So apoptosis is an enzyme mediated pathway. Okay, and it is highly regulated, while the necrosis does not involve any kind of caspases. Okay, so caspases are involved only in apoptosis but not in necrosis. And again, in the apoptosis, as I discussed before, the apoptotic bodies, they are engulfed by phagocytes. So there will be no inflammation. But unlike apoptosis, in the necrosis, what happens is that necrosis is a pathological response. Okay, so this is a pathological procedure which happens when cell undergoes injury. So let's say that there's some wound and the cell has disrupted beyond, and it has damaged itself beyond the repair. In such cases, or let's say there's some cytotoxic activity, there's some stress, like cell is in some environment which is not feasible for that cell to grow. In such cases the cell chooses to die, and that is what we call necrosis. So when this happens, the plasma membrane is disrupted. Okay, so unlike apoptosis, I told you in the apoptotic bleeding there will be no disruption of plasma membrane, but in necrosis the plasma membrane is disrupted, which leads to the spilling of subcellular components, like spilling of heat shock proteins, then DNA of that cell and other regulatory proteins in the surrounding tissue. So all of those subcellular components will be spilled in the surrounding tissue, which will cause in the activation of inflammasome. So the in necrosis, the inflammasome is activated, and this activated inflammasome it will release the cytokine factors such as interleukin, IL-1 beta. Okay, so and which will lead to the inflammation. So necrosis is always associated with inflammation. So if there is necrosis, there is injury which causes cell death. Uh, necrosis is the cell death which is caused due to injury, while apoptosis is the cell death which is programmed by the cell itself. Okay, maybe due to the genomic instability, due to some DNA damage, or the cell is under stress. Due to that, apoptosis happens. While necrosis is due to the injury to the cell, which is beyond control, which is due to which the plasma membrane gets disrupted. So these are very different phenomena that happens. Apoptosis can also, it is very important phenomena in development because it leads to digit formation. So apoptosis is also used when you have to remove unwanted cells. Okay, so this is not just happens when you have to remove like this, when you have to remove the genomic instability from the environment, like from the intracellular environment, but you, whenever there's any kind of unwanted cell during the development as well, apoptosis happens. For example, during development, the hands, the hands are something like this, so but you have to form the digit, so digit should be something like this, right? Okay, so digit should be something like this. So this area, this portion of cells, this is unwanted, so those cells are unwanted. Okay, so those cells will be killed by the apoptotic mechanisms. Okay, so whenever there are unwanted cell, so the cell could be unwanted due to multiple reasons. Maybe it has damaged DNA, maybe it is migrating abnormally, maybe is it has no significance on that place during the development. So in all those circumstances, the cell chooses for the apoptotic mechanisms. So I hope the difference between necrosis and apoptosis is quite clear to you all. And by now you must have got what is apoptosis and why do cell exhibit apoptosis. So now let's learn about the apoptotic mechanisms, the pathways for apoptosis. So there are two types of pathways for apoptosis. First one is intrinsic pathway and other is extrinsic pathway. Okay, both of them have their own significance. The intrinsic pathway is followed when there is intracellular signal that now the cell needs to carry the apoptosis. The intracellular signal could be maybe the DNA damage, okay, like double-stranded DNA breaks, or it could be due to oxidative stress or hypoxia, the low availability of oxygen, or due to some cytotoxic drugs. Okay, all of them, they are going to elicit as an intracellular signal that the cell needs to undergo apoptosis. Now the cell has the DNA damage which cannot be repaired, so the cell, instead of accumulating all that genomic instability, and instead of disrupting the genomic integrity, let's just kill that cell. So whenever this happens, it's the cell senses intracellular signal. So the intracellular signal that the DNA is damaged, or that there is some stress under which the cell cannot survive. So it is better to let it die than to continue the survival, which can lead to genomic instability. So at such condition, this is sensed by the proteins of BCL2 family. So BCL2 family proteins sense this intracellular signal. Okay, and then the intrinsic pathway starts. Okay, so intrinsic pathway is always elicited after the intracellular signal, while the extrinsic pathway is due to the extracellular signals. The extracellular signals that come from the cells which are like the neighboring cells. So they are the receptor mediated pathways. So unlike intrinsic pathway where the inner damage, the damage that has been done inside the cell, within the cell, that is sensed by some proteins, unlike that pathway, in the extrinsic pathway there are some receptors on the cell. Okay, those receptors are known as death receptors. Okay, so those death receptors, they are the ones which are going to receive the extracellular signals. So extracellular signals, obviously here they will be ligands. So those ligands are known as death ligands. Okay, and those death ligands will come and they will be sensed by the death receptors. So this is extrinsic pathway is receptor mediated, while the intrinsic pathway, that will be the intracellular signal in that intrinsic pathway, that will be sensed by the BCL2 family proteins. So those BCL2 family proteins, they are of two types. Okay, the the pro-apoptotic proteins, while there are some anti-apoptotic proteins. As the name suggests, the pro-apoptotic proteins, they are promoting the apoptosis. So they are the ones which are going to promote the apoptosis by the release of cytochrome C. So this intrinsic pathway, this is mitochondria mediated. Okay, how we will see that in short while. This is mitochondria mediated pathway, while this is a receptor mediated pathway. Extrinsic pathway is receptor mediated. So the BCL2 family proteins are of two types, the pro-apoptotic proteins and anti-apoptotic proteins. Let me write the examples here. So the BAX, BAK, BAD, BCLXS, then BID. BID is a quite important one. I'll discuss that. BIK, BIM, HRK, all those, they are pro-apoptotic proteins. Means whenever there will be aggregation of those proteins, they will lead to apoptosis. Well, anti-apoptotic proteins are the one which will, which will keep a check on the apoptosis, which will inhibit apoptosis. BCLXL, so the BCL2, BCLXL, BCL MCL1, those are the ones which are anti-apoptotic proteins. You do not really need to remember all those names, but remember there are quite some important ones like BAX and BID. Both of them, the pro-apoptotic proteins are important to remember, and BCL2 is anti-apoptotic protein. So even if you remember those three, that is more than sufficient. Okay, so these are the apoptotic proteins. Some of them promote the apoptosis, while some of them inhibit the apoptosis. The pro-apoptotic proteins, they support the apoptosis by promoting release of cytochrome C. I'm sure you have heard the name cytochrome C. So cytochrome C, it is involved in the electron transport chain reaction. So it is a mitochondrial protein. So this cytochrome C, it is very important in mitochondria for ATP production because it is a part of electron transport chain reaction. So this is meant to be present on the membrane of mitochondria. But when this is released inside the cytosol, so cytochrome C will be released from mitochondrial membrane to the cytoplasm. Okay, and when it is released in cytosol, this is the thing which is which is like pointing out that there is something wrong with the cell. So the cytochrome C is meant to be on the mitochondrial membrane, but when it gets released into the cytosol, sends a signal to the cell that something is going wrong and it needs to undergo it needs to kill itself. So that is why the pro-apoptotic factors, they promote the release of cytochrome C, while anti-apoptotic factors, they inhibit cytochrome C release. Okay. So this is how they inhibit the apoptotic pathway. So let's see what downstream signaling happens when the cytochrome C is released. Okay, let me just, we will come to this figure later. Okay, better. So in the intrinsic pathway, there are some factors that we now see like listed down. So the BCL2 family proteins, which has some pro-apoptotic proteins such as BAX, BAK. Okay, those are the ones which are going to promote the mitochondrial release, mitochondrial protein release, and let's write the name of the protein. So they will promote the cytochrome C release. Okay, so this is the very first step. So once the cytochrome C is released in the cytosol, it goes and binds to APAF1. It goes and binds to this protein. This is known as apoptotic protease activating factor. So APAF1 to cytochrome C goes and binds to APAF1. So it forms a multi-protein complex. So cytochrome C plus APAF1, this multi-protein complex is called as apoptosome. Apoptosome. Okay, so this apoptosome, what is apoptosome? The complex, the multi-protein complex of cytochrome C and APAF1. This is going to recruit. This is going to act as a platform. So this acts as a platform, and it recruits procaspase 9. Okay, this is very important. So it recruits procaspase 9 via CARDs. Okay, what is this? This is CARD is the caspase activation and recruitment domain. So via the caspase activation and recruitment domains, this apoptosome, which acts as a platform, it recruits the procaspase 9. So procaspase 9, this is an proenzyme. Okay. So proenzymes are the inactive form of the enzymes. So this proenzymes get cleaved. Okay. So it undergoes the partial proteolysis. So it gets cleaved, and after undergoing the partial proteolysis, partial digestion, it gets converted into caspase 9. Okay, so this is going to start the downstream signaling. So this is going to initiate the downstream signaling, and that is why procaspase 9 is also called as initiator caspase. Okay, this is called initiator caspase. This is initiator caspase, which is going to start the downstream signaling and which is going to then activate the executioner caspases. Okay, so what happens is that the caspase 9 proenzyme, which is now recruited to the apoptosome, this case gets cleaved and gets activated. So this is an active form. Okay, this is an active form of the enzyme. So caspase 9, once it gets activated, it recruits the executioner caspases. So executioner caspases are caspase 3, caspase 6, caspase 7. Okay, so these are the executioner caspases which are going to do the proteolysis. So they were also present in the inactive form in the cytosol. So they were also present in the proenzymatic form, and then they get cleaved. Like they also undergo partial digestion with the due to the caspase 9. So this is the initiator caspase, while these are the executioner caspases. Now those are the caspases which are going to degrade all of the proteins. So they degrade nuclear lamins. Okay, so due to which the whatever nuclear contents will be there, they will be spilled out. Then in the cytosol, obviously, then they are going to break down the cytoskeletal components. Okay, so basically they are the proteases. So they are going to break down the all of the proteins. So they will break down the structural proteins, the regulatory proteins, and there's a very important one known as ICAD, the inhibitor of caspase activated DNAs. Okay, so this inhibitor of caspase activated DNase, ICAD, this will also be digested by the caspases. So this is an inhibitor of a DNase. Okay, so what does DNase do? DNase, it cleaves the DNA. That is why the A is like it is going to cleave the DNA. The substrate that this enzyme works on is the DNA. So this DNase normally shouldn't be working, right? And that is why the inhibitor of caspase activated DNase, this keeps like this inhibits the DNA so that DNA will remain intact. But now this inhibitor has been broken down by the help of caspase. So this ICAD will be converted into CAD, the caspase activated DNase. So this caspase activated DNase, this will break down the DNA. So this will do the DNA fragmentation. Okay, so this is how a caspase, which is a protease, so this is a protease, so that is why it will break down the protein which is going to like which was protecting the DNA, and that is so now the protector of DNA is gone due to this caspase, due to this protease. Now DNA will be fragmented. Okay, so this leads to the DNA fragmentation. So once the DNA gets fragmented, once all of the proteins get fragmented, the cell blebbing happens, and then it forms apoptotic bodies, which will be engulfed by the phagocytes. So this is how a cell will be killed. A cell will perish very clean. Like this is a very clean process because it won't elicit any kind of immune response. It won't provoke immune system to exhibit the inflammation. So that is why this is a very regulated process. This is a tightly regulated process. So this was the intrinsic pathway. The intrinsic pathway is mitochondria mediated. Okay, let's now take a look at the extrinsic pathway. So the extrinsic pathway is a receptor mediated pathway. There it won't directly release the mitochondrial protein, but it does ensure that whenever extrinsic pathway is activated, it even activates the intrinsic pathway. We'll see how. Okay, let's first learn about the extrinsic pathway. So what happens is that the cells have some receptors called the death receptors, which react to the death ligands. So the death ligands will be released by the neighboring cells. Okay, so the death ligands are the fast associated ligands, okay, or the TNF alpha. Okay, so those are the death ligands. While the death receptors, which are present on the cells, they are FAS. Okay, the FAS CD95, or the TNF receptor 1. So those are the death receptors. So those death receptors have a death domain on the cytosolic side, okay, to which the adapter protein like FADD, this is FAS associated death domain. This is FAS associated death domain. And then there is TRADD, TNF receptor associated death domain. So these are the death domain which are present on the cytosolic side. So these are the adapter proteins to which the proenzymes, the procaspases 8 and 9. In this case, there were the initiator caspase was 9. In this case, the procaspases are 8 and 10. Procaspase 8 and 10 can be recruited. So on those FADD and TRADD, those procaspases can be recruited, and this forms a signal known as DISC. Okay, sorry, this forms a signaling complex, not a signal. This forms a complex known as DISC. DISC is death inducing signaling complex. So this complex has quite a literal name, death inducing signaling complex. So what is death inducing signaling complex? It is the complex formed by the death receptors, and their death domain on the cytosolic side, as well as the proenzymes. So once this death-inducing signaling complex is formed, it is going to do the downstream signaling, which is going to lead to the apoptosis. So let's summarize all of the process through schematic. So this is a schematic which was given in this particular research paper. So this is a review article. If you want, I can put the PMID or the DOI of that review article in the description box so that you can refer to this article. So I have taken this schematic from that article. So here you can see that it is summarizing both of the pathways, the intrinsic pathway as well as the extrinsic pathway of the apoptosis. So let's first discuss the extrinsic pathway. Here you can see the ligand, the death ligand like FAS-L, okay, or the TNF alpha. They can come and bind to the death receptor, and the death receptor has the cytosolic domain, the death domain known as FADD or TRADD, the FAS associated death domain, or the TNF receptor associated death domain. So this death domain is going to recruit the procaspase 8 or 10, and this whole complex, the receptor, the death domain, and the procaspase, the proenzyme, this complex is known as death inducing signaling complex, which is going to do the downstream signaling. So this procaspase will be cleaved. Okay, so this will be cleaved, and this after the partial digestion, it will form the caspase 8 or caspase 10, and this caspase 8 and caspase 10, those are this is initiator caspase in this scenario. This will cleave the procaspase 367 and will convert it. So this is the inactive form, and this will convert it into the active caspase 367, which is going to do the cleavage of the cellular substrate. And this is how the apoptosis is led by the extrinsic pathway. So both of the pathways, the intrinsic and extrinsic pathway, they converge at the procaspase 367 cleavage. So in both of the cases, the caspase 367 are the ones which are going to execute the cleavage processes. So that is why both of the pathways, they converge at the procaspase, the proenzymes of the executioner caspases. So let's summarize the intrinsic pathway once. Okay, so the BCL2 family proteins, they are the ones which are going to receive receive the intracellular signal. So whenever the cell is, it is suspended to stress. So wherever there is stress, like oxidative stress or hypoxia, the BCL2 family proteins, they will be activated. So the anti-apoptotic factors will be suppressed, while the pro-apoptotic factors will be activated. Like the expression of the pro-apoptotic factors will be more. The backs and back, they will undergo oligomerization. So which will lead to the mitochondrial release of the cytochrome C. So here you can see the mitochondria releases its protein cytochrome C, which binds to the APAF1. Okay, APAF1 is the apoptotic protease activating factor 1. So once it binds to APAF1, this forms the apoptosome. And this apoptosome is the APAF1, cytochrome C, and the procaspase 9. So this procaspase 9 will be then partially cleaved, and it will get activated. And once it gets activated, it activates the proenzymes of 367. Okay, so I hope this whole pathway is clear to you all. So I also mentioned that the extrinsic pathway, it is going to activate the intrinsic pathway as well. So intrinsic pathway will be activated on the two conditions. First, there is some intracellular signal that the DNA is damaged or whatever. The intracellular signal can start the intrinsic pathway, but extrinsic pathway can also start the intrinsic pathway. Let's see how. So what happens is that whenever the death ligand comes, it activates the death receptor and activates the procaspases 8 and 10. The caspase 8 and 10, it has a property to cleave one BCL2 BCL2 family protein, and that BCL2 family protein is BID. And while explaining the pro-apoptotic factor, I told you that remember this, because this is an important one, because the BID This is the one which is helping us to have a crosstalk between intrinsic pathway and extrinsic pathway. So the caspase 8 cleaves the bid, this pro-apoptotic factor, and after cleavage it gets converted into T bids. This is truncated bid. Okay, the truncated bid. T bead. So this T bead, this permeabilizes the mitochondria, and once it permeabilizes the mitochondria, the cytochrome C will be released and the intrinsic pathway will be followed. So this ensures that once the extrinsic pathway, like once this cell, the neighboring cell, sends the ligand to perform the apoptosis to the cell, this ensures that the alongside the intrinsic pathway, extrinsic pathway also starts, so that it will like the apoptosis, like apoptotic mechanism will be faster. So it will ensure that the cell will undergo apoptosis. So for increasing the efficiency of the mechanism, extrinsic pathway also starts the intrinsic pathway. That also mediate, like also mediate the mitochondrial release of the cytochrome C. So with this, we have covered all of the important details about the apoptotic pathway. In the next lecture, we will see how this apoptosis is, it is avoided, so that the cell will not die and the tumor cell will continue to have uncontrollable growth. Till then, see you. Bye bye.